Hello students, good morning. So in this video, I will explain you paper 4, biology paper 4, October-November series 2018. So let us start discussion on this paper. Question number 1. Let us discuss question number 1. Wetlands are important ecosystems. Researchers studied the feeding relationship between the organism in an area of wetland on the coast of Texas. Figure 1.1 shows part of the food web. So student, what is food web? Food web is the network of interconnecting interconnecting food chains. So like you can see in the figure 1.1, you can see in the figure 1.1, right? So many food chains are there. How many food chains? Let us see. 1, 2, 3, this is 4, this is 5, this is 6, this is 7, this is 8, and this is 9. 9 food chains are there which are interconnected to form this food web. So at the bottom of this food web, you can see here like clover, grass, algae, and phytoplankton. These are producers that make food for other organisms. So this is the first trophic level, right? So these are producers and rest are consumers. Let us see like pipefish, water fleas, oysters, snow goose, muskrat, right? So these all are primary consumer, right? They form second trophic level because they are feeding directly on producers or first trophic level organisms. So let us see, students, discuss about it further. Question number 1, A part. Complete table 1.1 by giving the name of one organism from the food web. In figure 1.1, for each row, producers, it is grass, you can write grass, clover, algae, and phytoplankton. So anyone can be written. Okay. Secondary consumers, bald eagle. Now secondary consumers. So we have to see what are secondary consumer over here. Primary consumer I have just shown you. So for food chain 1, Blaney is the secondary consumer, or it forms third trophic level organism. For food chain 2, mysid shrimp is what? Secondary consumer. And for food chain 3, stone crab is secondary consumer, right? And for food chain 4, eagle, bald eagle is the secondary consumer for this secondary consumer, or T3 tropic level, this one T2 and this T3, okay, or secondary consumer. Okay, similarly we can see for other food chains, say if we are talking about the grass, right? Grass is being eaten up by snow goose, this is the primary consumer, and bald eagle is secondary consumer for it. Similarly, we can see food chain 5th, right? Muskrat is what the primary consumer, and bald eagle is secondary consumer. If we talk about the food chain 7th, okay, here bald eagle is secondary consumer, and for 8th food chain, right, this marsh rice, it is the secondary consumer. So, students, anyone you can write bald eagle, you can write marsh rice rat, you can write stone crab, mysid shrimp, or blenny. Anyone you can write. So I have mentioned here bald eagle, an animal that feeds at 2 tropic levels. So here you can see this blenny. Blenny is feeding on mysid shrimp. This is tropic level 3, as well as it is feeding on pipefish, that is tropic level 2. So we can write blenny also, or we can write here bald eagle. We can write as bald eagle is feeding on bald eagle is, for this it is the 4th tropic level. It is feeding on uh, 3rd tropic level as well as on 2nd tropic level. This is 2nd tropic level. So anyone can be written. Okay, now the functioning of ecosystems relies on the cycling of nutrients. Figure 1.2 shows part of the nitrogen cycle. Part of nitrogen cycle. So let us discuss about it. The ammonium ions. Ammonium ions are converted to nitrate ion. So who is responsible for this? A. Process A. This is called nitrifications. Nitrification. And nitrogen gas in the air is okay, fixed as nitrate ion. So this is called nitrogen fixations. Nitrogen fixations. This process is done by leguminous bacteria that is found. Say this is a leguminous plant. These are the root. Okay, in the root nodules there are nodules. Okay, so here that bacteria harbors. This bacteria is capable of absorbing atmospheric nitrogen. Its name is rhizobium. So, and nitrate ion are moving to plants. By plants have root hairs, they absorb nitrate ion by a process called active transport. Active transport. As the nitrate ions are low in concentration in the soil with respect to plant root cells. So plants have the root hairs. So that root hairs basically they provide large surface area for efficient absorptions, and they have some characteristic like a root hair cell have a thin cell wall. Okay, and in the membrane there is transport proteins that help in transporting the nitrate ion from the soil into the root. Since it is happening against the concentration gradient, so there is use of energy produced by root cells by aerobic respirations. Here in plants, amino acid is converted to, this nitrate is converted to amino acids by, nitrate combines with glucose to form amino acids. Amino acids now is converted to protein in the process D, in the part of the cell D. So in the cell, plant cell, there is a ribosome. In the ribosome or rough endoplasmic reticulum. These structures are found in the cytoplasm, okay, where the amino acids are assembled in a particular sequence to make proteins. Let us see here. So, students, you can see here state the name of the process A. This is nitrifications. I've already explained. Type of organism is bacteria, soil bacteria, rhizobium, anyone. You can write part C. Describe how nitrate ions enter the root of plant, shown by arrow C. So basically, the plants, they have the root hairs. The root hairs are there. Okay. This is, I'm drawing here. You can see this is the root hair. There are many root hairs. They provide large surface area. You can see in this root hair there is a large vacuole. Okay, and the cell wall is thin, right? So from the soil, the minerals, they can diffuse. Minerals can diffuse across this membrane into the root hair. Okay, by active transport. So here you can see nitrate ion diffuse from soil to root across cell membrane by active transport. Okay, cell membrane contains this cell membrane. This cell membrane contains this transport proteins across which the minerals are to be transported from their low concentration area to high concentration area, right? Using energy. This energy is produced during aerobic respiration of root hair cells. State the name of the structure in plant cells where process D occurs. Process D is the synthesis of protein from amino acids. Basically, the synthesis of protein from amino acids, right? It is done in the cytoplasm on ribosomes or rough endoplasmic reticulum. Rough endoplasmic reticulum is also found in the cytoplasm. It has it has uh, ribosomes embedded on its surface where the protein synthesis occurs. Amino acids are combined each other by chemical bond to produce this polymer protein. So you can write the name of the structure. Okay, name the process that occurs at B, that is nitrogen fixations. Okay, a pyramid of numbers of wetland ecosystem showed that there were very large numbers of organisms at the base of the pyramid and a very few at the top. So, student, you can recall ecological pyramids. Basically, ecological pyramids are graphical representations of various trophic levels of food chain. So you can see at the base it is broader. As we move towards apex, it tapers. Okay, number of organisms reduces. Least number of organisms here. Okay, here most number of organisms at the base. Why it is so? So students, when energy is transferred from one trophic level to next, the amount of energy transferred is very less. That is around 10% only. 10% energy is transferred. Most of the energy is lost. That is 90% is lost either as heat or in metabolic process like respirations or in movement or in some other activities or maybe means whole food is not being digested. The animal is also excrete, right, waste. So in that also energy is lost. So only 10% is passing from one trophic level to another. Basically, the bottom bottom is formed by producers. So there are more producers, green plants at the bottom, and they make food by the process of photosynthesis, right? So this is first trophic level, and so number of plants are more. And second trophic level is primary consumer. Okay, so that is for the less number of organism, right, which can be supported by the producers. And then further, now these primary consumers will be eaten up by secondary consumer or a third trophic level organism. Here again, the 10% energy will be reaching there, so number will further reduce. Okay, so amount of energy reaching at the top trophic level, okay, is less, not sufficient to support the many organisms. Therefore, there are less number of organisms at the top. Question number 2, students. Now, question number 2. Uh, the figure 2.1 shows a dwarf sunflower. You can see dwarf sunflower and a tall sunflower. The height of the dwarf sunflower is here 0.45 meter, and for the tall sunflower is 4.5 meter. Now, the dwarf plant like the one in figure 2.1 have mutant allele. What are mutant allele? Allele or gene, right, whose base sequence is changed. Okay, they have changed base sequence. So, what is allele? Mutant means changed. Change base sequence in the gene. So, what is allele? Allele refers to alternative form of a gene. What is it? Alternative form of a gene. Say a characteristics called height. Since here dwarf and tall plants are there, so they are representing height. This is one of the characteristic feature trait. Now this feature is can be either dwarf. A person can be either dwarf or tall, and this height is controlled by genes. So there are two forms of the gene because gene is controlling height. So the gene controlling height or allele, the alternative form of a gene, that is here say capital T, capital T. This is controlling height, or it could be capital T, small t, or it could be small t, small t. So you can say the genes can be alleles. Alleles. So two types of alleles are there. One is dominant, other is recessive. So dominant allele is controlling tallness. This is capital T, and genes are alleles are found in pairs. Okay, so either it is in pure form or it is in impure or heterozygous state, mismatched. You can say lowercase t, okay, and uppercase t. Lowercase t, remember, it always control the dwarfness, lowercase t. So this is recessive. This is recessive allele, and uppercase T is dominant allele. This is dominant allele. Dominant allele. Okay, so you can see here in presence of dominant means that it can express in pure state as well as in hybrid state, right? So wherever the dominant allele will be there, it will be able to express. So tall plants can have the pure or hybrid alleles, okay? And dwarf plants, the plant will be dwarf if both the alleles are recessive. Only in pure form it can express. So this alternative form of a gene is called allele. So here B part, let us see. Shoot growth in plants is controlled by auxins. What is auxin? Auxin is a plant hormone. The meaning of auxin is to grow. To grow. This hormone is produced. This hormone is produced at the tip of the root or shoot. Tip of the shoot or root. So an enzyme in the shoot tip converts molecule of amino acids into auxin. So this is an enzyme control process. Explain how a mutation. What is the meaning of mutation? Mutation means change in DNA results in the abnormal enzyme, which does not catalyze the reactions in figure 2.2 here. So let us see, mutations causes change in base sequence of DNA. Say this is the one strand of DNA. I'll draw this is one strand of DNA. It has a base sequence A, C, T. Other strand which is complementary, sequence A pairs with uh, A pairs with T and C pairs with G and T pairs with A. So by 2 hydrogen bond, C by 3 hydrogen bond, 2 hydrogen bond. You can recall, students, we discussed about this in chapter 4, right? So this is the base sequence. So what happens in mutations? What happens in when it gets mutated? So base sequence is changed. Say this is changing. This sequence is changing here. So what is happening in the new DNA or mutated DNA? We find in place of this there is C, there is A, there is T, right? So here C G, and this is T, and this is A. So you can see now sequence position of base sequence is changed here. So it will result in abnormal protein, a different protein here. Sequence of amino acids will be different in proteins. Okay, so mutations causes change in base sequence of DNA, number one, and DNA codes for protein. Remember, students, so DNA basically it contains information in the form of codon. There are 4 nitrogen bases are found in the DNA molecule, that is A, C, T, G, right? So these 4 bases only, right, controls everything. So in the form of codon, the information is present in DNA for protein, right? So but DNA cannot come out in the cytoplasm because it can be broken down by the enzymes or chemicals found in cytoplasm. Okay, so it needs to be protected. So it passes its informations through mRNA. As the name indicates, M is messenger. This brings message in the form of codon from DNA. This is called transcriptions. Right? And this messenger RNA carry these informations to the cytoplasm, and here on the ribosome. This is the ribosomes here. This mRNA on the ribosomes will translate. This is called translations. The information present in RNA will be used to make protein. So here the protein molecules will be made. This is what protein molecules. So this protein can be used to make enzymes. So here what will happen? DNA codes for since DNA codes for protein through mRNA, so different amino acid sequence in enzyme will change the shape of the active site. Say this is the enzyme, you can see, and this is the shape of the active site. This is enzyme and this is shape of active site. Active site. What is active site? A place where the substrate will attach. So what will happen? Okay, in abnormal enzyme, then the protein is abnormal, where the sequence of amino acid is different. Then what will happen here? Now, due to different sequence of amino acids, now shape will change. This is a change shape, right? So here now a substrate molecule cannot fix. This was a substrate, right? This can fit here, but this is what substrate. This cannot fit here, but here only it can fix. So the reactions will not take place. This is very important concept, students. You need to remember and understand. Now, two tall sunflower were crossed, and 25% of offspring produced were dwarf. Explain how it is possible for two tall parent plant to have this percentage of dwarf offspring. Number one, you should remember the dwarfness is controlled by recessive allele. Recessive allele, I just told you that this is lowercase t, it is the allele, okay, that controls dwarfness. And this is always in pair. If it is in homozygous, then it will. And both, second thing is what? Both the plants are heterozygous. If you see the genotype of both the plants, it is heterozygous. This is genotype of one plant and this is genotype of another plant. Both are heterozygous. D part, let us see now about this. Show how several strawberry plants can be formed from one parent plant. Here you can see, students, in this diagram, in this image, that it is basically cutting. They are cutting branches of the parent plant, and putting it in the pots, soil present in pots, right? So only one parent plant is involved in this kind of reproductions. So it is explain the type of reproduction that produces plants by this method shown in 2.3. The type of reproduction is asexual because one parent is involved, okay, and cells of the growing offsprings, they will divide by mitosis, cell division, okay, and it will produce all the offsprings, right? Offsprings, they will have genetically similar type of cells. Genetically identical cells will be produced. So plants will have the same structure, same shape, same requirement. Explain the disadvantage of the type of reproductions. What is the disadvantage of such type of reproductions? Like since the offsprings, they carry the same type of cells, same genes, okay, so there is no or very little variations. Because of this, they are not able to respond properly if there is any change in the environmental conditions. For example, say the parent plant is able to live uh, nicely at the low temperature. The offspring will also do, because same genes they are carrying. Okay, and if there is change in the environmental temperature, it becomes very hot. So the offspring will not be able to survive. Second point, uh, since they have same genes, they have come from one plant only, same plant only, so they will have the same type of requirement. So they will be feeding on the same type of resource. As a result, there will be more competitions for that resource, and they can there will be more risk of inheriting harmful traits. If the parents have some disease, right, same disease will be passed on to their offsprings also. So more risk of inheriting harmful traits. And there will be, of course, no evolutions, because they are carrying the same genes. There is very little or no variations. Okay, so no new species will be formed. Question number 3, students. Figure 3.1 is a photomicrograph of some xylem vessels. You can see the xylem vessels. The wall of the xylem vessel is thick in this photomicrograph. Okay, it is hollow, having more space, right? So state one structural feature. State one structural feature, okay, of xylem vessels, and explain how this is related to functions of water transport. You know, they have thick cell wall, so they can withstand water pressure. Beside this, you can write some other also, because they are forming a long hollow tube. They have no living materials. There is no cross walls. There is no cross walls, right? So more water can flow without any obstructions, since their work is to transport water from root to the leaf. So water can, without any hindrance, any obstructions, it can flow. Explain the mechanism that is responsible for the movement of water in xylem vessels. Mechanism. How the water is moving in the xylem vessels, that I just so loss of water as vapor from the mesophyll cells through stomata. As a result, what will happen? It will cause low water potentials in the leaf cells. Say this is a leaf. Okay, so from here, what happens? The water is lost as vapor, right? So through stomata, it will diffuse out. Now, the cells of the leaf, right? They will have low water pressure, so water potential will decrease, and this will pass on to stem. This is stem. This will pass on to stem also. Okay, this tension will pass on to stem. This is called transpiration pull or suction force. Since there is less water, okay, water pressure in the leaf cells and in the stem cells also nearby. So water in the stem, in the xylem, we can say, in the xylem, right? It forms a continuous column, because between water molecules there is a force called adhesion or cohesion force. Cohesion force is acting between water molecules. Cohesion force. Okay, so this keep them in one continuous water column. This bind them. Okay, and when there is a suction force, or you can say transmission pull is created, okay, in the leaf, and comes in and the stem, it will pull this water column up to the leaf. So this way water can move. So from one cell to another, water is moving by osmosis. Okay, and this water as a column can be lifted up because of the diffusion of water as vapor through stomata. So what is important here, students, you need to remember that loss of water as vapor from mesophyll cells through stomata, okay, and lowering of water potential or less water pressure in the leaf, that will create suction force or transpiration pull. Water molecules are bind together by cohesive force, forming a continuous water column. Okay, and because of the loss of water from the leaf, right, this water column will be lifted up. Water column will be lifted upward to leaf. So this you can write down. So more transpirations, the more fast water will be lifted up. Now, third part, state one role of the xylem vessels other than transport. Since the xylem vessels, wall of the xylem vessels, it is having a deposition of chemicals like lignin, or sovereign. You need not to remember the name. These chemicals are there, right? So that makes the wall strong. So it provides support. Next, the rate of transpiration is affected by several factors, including temperature and humidity. State and explain the effect of an increase in temperature on rate of transpiration. Rate of transpiration can increase or decrease when there is increase in temperature. How it happens? Let's see. So when the temperature increases, so water molecules, their kinetic energy increases. Okay, diffusion of water molecule increases. So as a result, the water molecules will diffuse in the guard cells from the cells of the leaf. Okay, when water molecules will diffuse into guard cells, what will happen? It will make stomatal pore wide. Stomatal pore wide, stomatal pore. So there will be more rate of transpiration. Transpiration will increase here. And due to more diffusion of water molecules, because the water molecules now moving out from here, so what will happen here? The guard cells, since from guard cells the water molecule are moved out because of a higher temperature, okay, so they will become flaccid. Here they were turgid and here become flaccid. Guard cells become flaccid, and this makes the stomatal pore narrower. Okay, so the rate of transpirations will decrease. So student, remember how increase in temperature can affect the rate of transpiration. It may increase or decrease the rate of transpirations by making the guard cells turgid or flaccid. Question number 4, students. Insulin is a hormone that regulates concentration of glucose in the blood. Insulin is a hormone. What is a hormone? Hormone basically a chemical substance produced by endocrine glands. Say this is a blood vessels, okay, and there is one endocrine gland. This is endocrine gland. This endocrine gland will pour its secretion directly into bloodstream. right? So blood will carry, say these are the hormones. These hormones are carried by the blood. Now these hormones will affect the activity of specific target organ. Why specific target organ? There are many organs are there, but it will affect only specific target organ, not this. Why? This only. Because they have the complementary receptors which have the similar shape as of hormone. So when you write the definition, you can write a chemical substance produced by endocrine glands. It is carried by blood and it affects the activity of target organ, specific target organ, remember. Now, next part. The two people, A and B, visited a doctor to discuss their similar symptoms. The doctor thought that their blood glucose concentrations were not very well controlled. A glucose tolerance test was carried out on both people. A and B did not eat or drink anything other than water for 8 hours before the test. They then drank a glucose solutions. Okay, blood samples were taken at 30 minutes interval. The samples were tested for glucose concentration. You can see in this graph it is shown, right? Glucose concentrations for at this after 30 minutes. After 30 minutes only, the sample was taken. Okay, glucose solutions was taken and then sample was taken for A and for B. So let us see the questions based on this. Question number, first part of this question is, use 4.1 to state the glucose concentration A and B at 180 minutes. 180 minutes from the graph we can see. So it is at 180 minute, right? For A and B, the concentration is different. Okay, for A at this point you can see, okay, when you will see it will come 88 here. Okay, small grid is equal to 4 units, so here this is 80. Okay, it is coming here, so this is 88 for B. For A it is at this point it is the peak. Okay, it will come here half grid, 2 full grid and 1 half grid, about 200, so 210 milligram, you can see milligram per 100 cc. Calculate the percentage increase in blood glucose concentration in person A between 60 and 90 minutes. So between 60 and 90 minutes we have to see in the graph first what is the concentrations, right? At 60 for A the concentration is this one. This is below 80. How many grid? 2 grids. So it is 72. Here it is coming for 72 at 60, and at 90 minutes you can see at 90 minutes will go all the way, and this is the concentration here. Okay, so it comes to what, 160 and 1 2 and half grid. Okay, up to this it is coming. We can stretch it up to this. Yes, so it is here 170. So we can see now the difference. So increase in blood glucose concentrations equal to 170 minus 72. How much it will come? It will come around 98. Okay, 98, and then we have to find out percentage. This is milligram per 100 cc. So percentage increase will be increase in concentrations, okay, 98 upon divided by initial concentration. Initial was 72, okay, at 60 minutes, so into 100. So when we calculate, it will come around 136.11. So we can round off it because they are asking about nearest whole number. So 136%. So this is a simple calculation, students. You can do it. You can take the data from the graph. Now, third part, describe how the response of A differs from response of B. So we can see for A, right, after taking the blood sugar, glucose solutions, okay, after 30 minutes it took, and then after 30 minutes blood sample was taken, it was observed there was sharp increase, okay, there was increase uh, in the level of blood glucose very fast, and it does not come back. If you compare with the B, right, there was increase, but it was slow increase, not a sharp increase. But here in case of A, it is steep increase, more sharply it is increasing, right. The peak is very high, whereas in case of B, peak is just simply you can see it is 100. Peak is 100, whereas in A, the peak is 210. And in B, it comes back to normal. It comes back to normal blood sugar level, whereas A does not come back to normal sugar level. So you can see the response, it increases more steeply and increases to higher concentration, that is 210 milligram per 100 cc, and the value, okay, blood sugar level does not return to normal in case of B. So these 3 points you can get from the graph and write down. Next part, explain the result of the glucose tolerant test shown by B. So increase in blood glucose concentration is detected by the pancreas in the beginning because there is slow increase, and insulin hormone is released into the blood by pancreas. It will stimulate the liver, right, or muscles, so that they can uptake the excess glucose from the blood and convert it into glycogen, so that then we can see the blood glucose concentration decreases to normal, and you can see in the graph, okay, in beginning after 30 minutes it increases slowly, and this increase in concentration is detected by pancreas. Okay, so pancreas releases hormone insulin. This hormone insulin stimulates livers or muscles so that they can take up the excess blood glucose and then convert it into glycogen. Slowly what happens? The blood glucose concentration comes to here. You can see this is normal. Now, fifth part, the doctor thought that person A had type 1 diabetes. So remember, type 1 diabetes are of two types, type 1 and type 2, right? Type 1 diabetes is also called insulin dependent dependent diabetes. In this case, pancreas, cells releasing insulin are totally malfunctioning. There is no production of insulin at all. So patient has to take insulin on a regular basis. So three symptoms were asked. We can write like frequent urinations, dry mouth, weight loss, blood vision. Any three can be written. Question number 5. Student, you can see this photo micrograph of human blood. Different blood cells are shown, two WBC and one red blood cell. So describe the difference in appearance, the appearance and role of these three blood vessels labeled. Like first, red blood cells. If you see appearance of this, it is biconcave in shape, right? Or it has no nucleus. You can see it has no nucleus, right? No nucleus is there. And the function, as you know, it transports oxygen, transport carbon dioxide, and phagocyte, if you see, it is larger than red blood cells. And the nucleus, it has irregular shaped nucleus or lobed nucleus. The function of phagocyte is to eat bacteria by phagocytosis. Next is lymphocyte. Lymphocyte, you can see, lymphocyte, they have large nucleus, okay, and they have a small amount of cytoplasm. They are smaller than phagocyte, right? So you can write like that, and their work is to produce antibodies. Figure 5.2 shows some of the stages of blood clotting. Okay, blood vessels, platelets collect at the break in the, okay, platelets release an enzyme. Okay, this is called thrombokinase basically, and the name of the enzyme is thrombokinase. This is not in the syllabus, right? So that converts prothrombin into thrombin. Now this thrombin, okay, will stimulate conversion of fibrinogen, that is insoluble. This is soluble protein, to fibrin, which is insoluble protein. This fibrin will trap the blood cells and form clot. So complete figure 5.2 by filling in the two empty boxes. So this I filled already. State two roles of blood clotting. First role is prevent blood loss. Okay, so and by forming a clot, and second, prevent entry of bacteria or pathogen in the body. Now C part, hemophilia is a sex-linked blood disorder in which blood takes a long time to clot. Hemophilia. Figure 5.1 is a pedigree diagram. This the pedigree diagram is given showing inheritance of hemophilia. So here you can see this is the parental generations or P generations or parental generation of first generation, second generation, and third generations. Okay, this is first generation, this is second generations, and this is third generations. So now key is given here. Male with normal clotting time. This is the white square, and black square, male with hemophilia, and white square, male with normal clotting time. Okay, and if you state the genotype of the people identified as PQR. Normal allele is represented by X uppercase H and mutant allele. Normal allele and mutant allele. mutant allele is represented by X lowercase h. So let us see. So here father is one of the parent, male parent is quite square. So that is X uppercase H Y and female P. Okay, so it must be carrier then X H because it is not affected. So on it is carrier. The allele mutant allele is found only on other X chromosome. So this is the genotype of male and female. Now for this in second generations first son, the genotype is it is affected, it is black square. So we can see X lowercase h Y and female it will be normal. So this Q son is getting married with a female. Okay, since all the children are normal, seems normal for this hemophilia. Okay, so the genotype of female will be normal. She will not be a carrier because male is affected. And genotype for R will be the son. So it has the normal genotype, normal allele. Okay. S daughter. Daughter will be because their children, daughter marries with T, their children, one of the child is affected. It means daughter will be carrier. So one of the X will have, X chromosome will have this mutant allele. And father T, whom she married, will have normal allele. Okay. So genotype of the children, we can see son is receiving X chromosome from mother, that is the mutant allele. Okay. Now, okay, so state the genotype of the people identified PQR. P is okay. P is X normal allele, X mutant allele. Okay, this is the genotype of mother of first generations, and the Q is the son. Okay, in second generations, that is affected one as it is receiving the mutant allele from mother. And R is the normal male child. So this is a very important concept students should remember. Then how to use, how to write the genotype. So as you know about male and female, about their sex chromosome, the male they have mismatched sex chromosome, that is XY, and female they have same type of chromosome XX, right? And mostly since they are writing it is sex linked, so the gene for hemophilia for mutant allele is always found on the X chromosome. So male will always be expressive because they have only one X chromosome. Female could be expressive or carrier, right? They are carrier, okay, they will not be showing the disease because of two X chromosomes, the mutant allele is found only on one of the X chromosome. If it is found on the both the X chromosome, then they will be hemophilic. The couple S and T are expecting another child. What is the probability that the child will have hemophilia? Okay, so first of all, we must know the genotype of S and T. It is given already. So S is a carrier and T is normal. So we can write S and T, S, S and T. S, the genotype is X, H, X, normal age. Okay, and T genotype are normal. Okay, so two types of gametes will be produced by S: gamete containing mutant allele, gamete containing normal allele for hemophilia. Okay, and T will also produce two types of gametes: gamete containing X chromosome, gamete containing Y chromosome. Two types of sperms are there. So when a cross is made, this will cross, mutant allele will cross with normal allele. Son will be normal. Here son will be this daughter will be so as you can see, when mutant allele interacts with normal allele of father, right, the child is affected. Similarly, we can see we can do the same thing for others also. This normal allele interacts with normal allele of father, then normal daughter, similarly with Y, and normal son. So this way we can see the probability of the child will have hemophilia is 25%. So we can see probability is just 25% only. 25%. Same thing we can represent by using Punnett square also. This is a good way to represent so here for S and for T parent and S parent. T parent is having the normal allele. You can see X, H, Y, and carrier parent has one normal allele and one mutant allele. So you can place it X, H, X, H, X, H, Y, X mutant allele, X normal allele, X mutant allele, Y. So from this graph, it is clear that only 25%, okay, 25% children will be having hemophilia. Define sex-linked characteristics. Gene is located on X chromosome. Characteristics are more common to male, as gene is located on sex chromosome. For this, right, the sex chromosome could be X. Mostly it is on the X or XX both. Okay, so mostly since the male has only one X chromosome, so the characteristics are more common to male. Question number 6 is the last question in this paper, students. Figure 6.1 shows Galapagos iguana, yes. So you can easily identify they are reptiles. Okay. So state two features that are used to classify animals such as Galapagos iguana as reptiles. Characteristic of reptiles you can write here like dry scaly skin, soft shelled eggs. State two features that are present in plant cell and are not present in cells of reptile. Cells of reptile means that is animal cell. Okay, so in plant cells, cell wall is present and chloroplast is present. You can also write permanent large vacuole. B part, Galapagos iguana feed on seaweed, which contains starch and other carbohydrate. State name of the enzyme that digests starch. Amylase. Amylase is the enzyme that digests starch. Starch to maltose. State the name of two parts of alimentary canal where starch is digested: mouth and small intestine. Small intestine, the digestive juice coming from pancreas containing pancreatic amylase, and in mouth there is a salivary gland that releases uh, salivary amylase or ptyalin to digest starch. There are many threats to wildlife in the Galapagos. Describe ways in which endangered species can be conserved. So any 5 points you can write, student, like protect habitats, ban on killing animals, law to protect species, create exclusive reserves, captive breeding, gene bank, removing alien species, reducing pollutions. Any 5 points you can write here. So one aim of conservation is to maintain resources in natural ecosystem. State three resources that natural ecosystem provide for humans: water, fuel like coal, oxygen, timber, wood, drugs, etc. So many things we get from the nature. So this was the last question, students. Thank you very much. Bye. See you.